All right, in this video, you are going to get a crash course in bioenergetics. Now, I've shown you in the past this diagram of world record speed versus distance. So the way I calculated this, I looked up the world record for the 10-kilometer run, so for the 10K, and divided the world record time by the distance, right? So we got meters divided by time, seconds, that's how we get speed. So we're looking at the world record speed for different events, from the 100 meter all the way to the full marathon. Now you might notice a couple of things while looking at this. Let me erase this. One is that it's not a linear relationship. I think most of us intuitively know that the further we have to go, the slower we have to be. All right, so we know that if we're going to run 100 meters, we would run this fast, and if we run a full marathon, we would run slower. And most of us operate under the assumption that the relationship between those 2 points is linear, that it goes down gradually. That if I go 10% farther, my speed should be 10% slower. If I go 20% further, my speed should be 20% slower. It doesn't work like that. It actually has this curvilinear relationship where our fastest speed is around the 100 meter, 200 meter sprints. And then we slow down quite a bit, going to the 400 meter and then the 800 meter and then the 1600 meter, like a mile, right? So I want you to look at this difference here. Here's the 100 meter and 200 meter world record speed. They're about the same. And now here's the 800 meter. They're only about 700 to 600 meters different, but the speed is way different. We're going from over 10 meters per second to about 8 meters per second. Now let's compare that to the 5K world record speed to the full marathon. These are like 35,000 meters different in length, but the average speed, it's very negligible. The difference is negligible. It's definitely different, but nothing compared to the difference between the speed during a 100-meter race and an 800-meter race. Now, the reason for this is all to do with bioenergetics, how we get our ATP. We can categorize the way we recycle ATP as either aerobic or anaerobic, and a lot of you are probably familiar with this idea. So, right, whenever we do muscle work, we can do a muscle contraction, we use ATP, and we break it down into ATP. As we've discussed in the past, we don't store a lot of ATP, so that means we have to be able to recycle it in order to do a subsequent contraction. And the way we recycle these is by metabolism, and we can categorize these processes as either being aerobic, requiring oxygen, or anaerobic, not requiring oxygen directly. Now, the faster you can recycle this, the faster you can run. So in events that are short enough to be fueled anaerobically, which is a very temporary thing, you can only go anaerobically for a little bit. In events that are that brief, you can recycle ATP very quickly, and that allows you to run very fast. But as soon as you go to longer events, where you're going to be going longer than you can recycle ATP anaerobically, now you have to shift down to aerobic metabolism, which goes through this recycling process more slowly, but it can keep going on for a long time. That's the reason why we have really fast sprints and then everything else is close to the same speed. So if we can categorize our exercises as being aerobic or anaerobic. Nothing's truly one or the other. You're always running both of these processes, but they predominate, right? So weightlifting is going to be when you lift a weight, like go bench press or power clean your max, that's going to be primarily anaerobically fueled. You're getting ATP in these anaerobic mechanisms. Whereas if you are running like a 100-meter, a 2 mile, anything like that, it's going to be largely aerobic. There will always be anaerobic components in these, but predominantly aerobic. And that dictates how quickly or what speed you can sustain. I'll explain a little bit more with the following graph or on another page. One more thing. So one of the differences between aerobic and anaerobic metabolism is where they occur. So this is a picture of skeletal muscle under a microscope. So you got all the actin and myosin in here. And then these little deals here, the circular things, are mitochondria. Okay. Now I say mitochondria, you immediately think to the one thing you remember from high school biology, the powerhouse of the cell. The mitochondria within the skeletal muscle, within all your cells, almost all the cells, that's where you're going to be recycling ATP aerobically. That's why we call it the powerhouse. It's where aerobic metabolism occurs. Anaerobic metabolism is going to occur outside of the mitochondria, closer to where the ATP is actually being consumed, and what we call the cytosol. Here's another view of it. Now we're looking at a cross section. So we got all the actin and myosin. Each dark dot over here would be a myosin, and then the gray in between would be actin that surrounds it. And then you can see the mitochondria weaving all around through here. So aerobic metabolism would happen in here, and anaerobic out here, closer to where it's needed. That's one of the reasons why anaerobic is faster. It's because it's happening right where the muscle contractions are occurring. Aerobic metabolism is much more complicated and requires a separate compartment to happen. And so that transit time for the ATP and all the ingredients is one of the reasons why it's slower. Now let's look at the relationship between how long we can sustain an exercise and the maximum power we can sustain. So if you want to run really, really fast, you can use the phosphogen system. Phosphogen is a direct transfer of a phosphate from one molecule like phosphocreatine to another. Hopefully you just didn't see that message that popped up on my screen. So the phosphogen system, since it's one step, we'll see it in a second, it allows for very rapid ATP resynthesis. So let's say that I want to run this fast. If I want to run this fast at that power output, I can do that by fueling the anaerobic system. Now, the problem with the anaerobic system is that you only have a limited store of phosphocreatine and ADP and these other things. And so it can only be sustained for a short amount of time. Now let's look at these others. You have another way to recycle ATP, and that's with anaerobic glycolysis, or the partial breakdown of glucose. It happens very quickly, and so you can get a lot of ATP per second. But if I want to run this fast again, the glycolytic system cannot recycle the ATP fast enough, and I cannot do it just with glycolysis. I would have to do phosphocreatine. So let's say I want to run that speed. We'll call this run 10 miles per hour. That's a power output, more or less. So if I want to run 10 miles per hour, I can run it that fast until my phosphocreatine is depleted, and then I'll have to slow down to something that can be fueled by my glycolysis at an ATP consumption rate that glycolysis can fuel. Now, the problem with glycolysis, it would be really nice if we could just sustain this for a really long time. But when you do it at maximum rates, at really fast rates of glycolysis, it tends to pollute the muscle with things like acid and phosphates. And those cause muscle fatigue, which would then cause you to slow down. So it's not a very sustainable way either. Now we have another way, aerobic metabolism of carbohydrates. Now it's going to be much slower. If I run out of my phosphogen system and then I go to glycolysis, and I run out of glycolysis, or the glycolysis is causing fatigue, I'm going to have to shift to a slower running speed with aerobic metabolism of carbohydrates. If all I had is aerobic metabolism of carbs, running 10 miles per hour up here would never be an option. I could not recycle the ATP fast enough, right? So aerobic metabolism can fuel a much slower rate of ATP usage, something down here, let's say. So if I go at that level, if I run at that speed, I can go fueled by aerobic metabolism of carbs for a long time. Generally, if you're going at a competitive speed in a race, you can fuel that speed for about 40 to 60 minutes with aerobic metabolism of carbs. And then you slow down or stop, usually because you run out of carbohydrates. Now, if you start to run out of carbohydrates, you have to shift down one more energy level to another form of aerobic metabolism, which is the aerobic metabolism of fats. Now, fats, everybody has a decent amount of fats, even if you're really skinny. It can fuel your exercise for a very long time. We're talking days. And so if you start to run out of carbohydrates, you're going to have to slow down again to a speed that you can fuel with just fats. Now, the fats and carbs are great because they don't really pollute the muscle if you do aerobic metabolism, whereas glycolysis and phosphocreatine, they're going to pollute the muscle and cause muscle fatigue. These anaerobic systems over here, these two are great for going fast, but they're not very good for sustaining for a long period of time. Now, that means that if you need to do something for a long period of time, like live, you need to have aerobic metabolism, oxygen facilitated breakdown of foods and recycling of ATP. So, do you see in this pattern here, we have the same general pattern as that the world record speed. See that curvilinear relationship? These bioenergetic pathways are what dictates that relationship. Now let's talk about why the phosphogen system is so transient. Why can't we sustain the phosphogen system for a long period of time? First off, let's talk why is it so fast? And then why can't we sustain it? The reason it's so fast is because it's one step and it happens right near where the muscle contraction is happening. So here's our actin and myosin that we talked about in the past. We break down ATP to do a muscle contraction, and now we're left with this ADP, right? So we have an ADP that is missing a phosphate. You got this phosphate hanging out here. And in order for it to be useful again, we have to get ATP back. We have to put that phosphate back on. Now, with an anaerobic system like phosphocreatine or adenylate kinase, we have a source of phosphate. So with the creatine kinase and the phosphocreatine, we have this creatine that carries a phosphate on it. We call it phosphocreatine. Yes, this is the creatine that people supplement for strength training, and we'll learn a lot more about that in class and in exercise physiology. So that creatine carries a phosphate, and what we're going to do is use this enzyme or little machine, creatine kinase, to rip the phosphate off and put it back on to form ATP. I guess I could write it this way. We're going to rip that phosphate off, put it on ADP, and now we have our ATP again. So it's one step, really fast. Now the issue, why we can't do it for a long time, is we only have a little bit of phosphocreatine stored, usually enough for about 30 seconds, 40 seconds of all-out exercise, maybe 20 to 40 seconds, depending on how fast you're going. Now, we have another system that's called the adenylate kinase system. I'll just write this here. So we have a different little machine, an enzyme called adenylate kinase. And that's going to take two ADPs, right? We broke down this ADP. So it's going to take those ADPs, it's going to rip the phosphate off of one and put it on the other. So now we have an ATP. So it's just another phosphate exchange. We take a phosphate off of one thing and directly put it back onto ADP to form our ATP, and we're back in business and can do another contraction. So being one step and happening right there next to where the muscle contraction occurs makes this ridiculously fast, but you only have a certain amount of that stored, so you can't do it for a long period of time. So why can't you, what about anaerobic glycolysis? So we have the phosphogen. We saw why it can go really fast, a lot of ATP per second, but we know you can't sustain it because you don't have a lot of it stored. But what about glycolysis? Why is it pretty fast and why can't you sustain it forever? So, glycolysis is the partial breakdown and rapid breakdown of glucose. Your glucose is a carbohydrate and it has six carbons on here. So that's six, right? And so we're going to go through this process of glycolysis. You'll learn all about it in biology, biochem, physiology. All we're going to do is we're going to take these carbons and rearrange them. And then as we pull them off one at a time and move them around, we get energy released to make ATP. Now, there's a ton of potential energy in these bonds between carbons. And glycolysis, we're rearranging these and modifying the bonds in order to get that energy out to put the phosphate back on ADP. Now, in anaerobic glycolysis, you take that six carbon glucose. So here's our six carbons. They form a circle in this case. And we're going to break them down to just a three carbon molecule, either pyruvate or lactate. And that gets us a decent amount of energy to recycle some ATP. And it's fast. It's about 10 steps. So we get a decent amount of energy out quickly, but there's still a ton of potential energy left in the pyruvate and lactate that if we just stop there, we miss out on. So the reason that it's fast or glycolysis can provide more ATP per second than the full processing of glucose is because you partially break it down. You only do the 10 steps instead of all 30 or 40 steps in the full breakdown. And that means you get a small amount of ATP. So it's usually 2 to 3 ATP for anaerobic glycolysis, but you do it in a fraction of a second, right? Whereas with aerobic metabolism, you'll see in a second, you'll get like 32 ATP somewhere in there, but it's going to take a lot of seconds. So in terms of ATP per second, anaerobic, the partial breakdown is much faster and provides more ATP per second, but it's not as sustainable. And the reason for that is as you do these partial breakdowns, you're going to produce a lot of acids in here. So like acid coming off all over the place here. And that acid and some other parts of it, phosphates, free radicals, other things are going to come off, and those cause muscle fatigue. So when you go to the full breakdown of carbohydrates, you slow things down to where you're producing the acids still, but you can buffer them so that they don't accumulate and cause muscle fatigue. Now, I'll dispel one rumor very quickly. You're in high school, you probably learned that lactate or lactic acid causes muscle fatigue. Uh, it's wrong. Lactate is just half of a glucose. It's half of a carbohydrate molecule. It's got tons of potential energy. More and more research is coming out every year showing that lactate is actually a great source of fuel and our bodies use it. It does not cause muscle soreness. It does not cause fatigue. Acid might, muscle damage does, but not lactate. And with learning this, we're actually learning how to use lactate in advantageous ways to treat different diseases and improve exercise performance. So you'll learn more about this in biochem and in exercise physiology, but get out of your head that lactate is bad. It's just partial glucose, and you can actually process it and get a lot of that energy out. So let's talk about aerobic metabolism now, the full breakdown of carbohydrates or fats. Now, to fully break it down, it takes many steps. So in way back here in creatine kinase, the phosphogen system, we were looking at one step. Easy to memorize, really fast to happen. When we get to carbohydrates, like partial breakdown of glucose with anaerobic glycolysis, it's about 10 steps, right? A lot more than one. So each of those steps takes time. And so it's the ATP per second is a little less. Now, with aerobic metabolism, we're looking at all of those steps of anaerobic glycolysis, all 10 of them, plus like 20 more, depending on how you break it down. So because it has more steps and you're fully processing that carb, that glucose, the ATP per second is going to be slower. You get a lot more ATP from aerobic metabolism. You'll learn you get about 32 to 36 ATP depending on the efficiency. So you get a lot of ATP out of that glucose, out of breaking down the glucose. You don't convert glucose into ATP. You just use the energy to recycle ATP. So you get a lot of ATP from that process, but it happens very slowly. And so that means that your recycling rate is going to be down here. Or with fats, the aerobic metabolism of fats, to process those fats, it takes even longer. We're looking at like 30, 40 steps. All this depends on how you break down the steps, but it's a slow process, but very efficient. You get tons of ATP out of it, uh, but it's slow. Now let's talk about aerobic metabolism. How are we actually getting ATP from aerobic metabolism? Well, you can call aerobic metabolism oxidative phosphorylation. The oxidative part refers to oxygen, and we'll talk about that in a second. The phosphorylation part refers to putting a phosphate back on ADP. So this is our mitochondria here. This is just a stereotypical simplified version of a mitochondria. They're much more complicated than looking like jelly beans, but this is how we usually illustrate it. So the mitochondria, these little things inside the muscle, on the inside of it, we have this enzyme or little molecular machine, an enzyme called ATP synthase. And on the inside of the mitochondria, it's going to take ADP and push it back together with ATP. Now, the way it does that is by having lots of protons up here, hydrogen ions. You have on the inside of this mitochondria, you have two walls. So you have the inner wall, the outer wall. Now, between those, we break down our carbs and our fats in order to make a huge stockpile of protons inside of this, in between the two walls. It makes a gradient, and I'll show you that in a second. But what happens with those protons is they flow down ATP synthase from an area of high concentration to low concentration. And as they flow down, they cause parts of ATP synthase machinery to move around and actually push the ATP and the phosphate back together again. They smush them back together. So the movement of the protons down their gradient, down ATP synthase, is what causes ATP to be recycled. Now, the use of oxygen and the carbohydrates and fats and all that, breaking down those are used to get the protons up here against their gradient. Now, let me use an example with a dam to help you understand how ATP synthase works. So you guys might remember I'm from Las Vegas, proud Las Vegan. We have the Hoover Dam in southern Nevada, and it is what provides power for southern Nevada, Arizona, parts of California. And the way it provides energy is with a gradient. Notice that we have lots of water on one side of the wall and only a little bit of water on the other side of the dam, right? Now, as the water goes down that gradient, it's going to spin turbines that generate electricity. So here's lots of water on one side and little water. So as the water goes down the gradient, it's going to come through here and spin something called a turbine. And as that turbine spins, it has a magnet in it, and that spinning of the magnet causes electricity to flow. And that's what's powering the homes in southern Nevada. So it's all about water going down a gradient. In the case of mitochondria, it's all about protons or hydrogen ions going down a gradient. So as they go down ATP synthase, it literally is causing part of the ATP synthase to spin, just like the turbine. And as it spins, it's rearranging the ADP and phosphate and putting them back together. So stuff going down a gradient causes spinning just like a turbine that then makes your ATP. So in the case of the Hoover Dam and any dam, the way we get water on one side versus the other is by having a lot of rain and snow upstream and not some downstream, right? We take advantage of the rain that like goes upstream and the river moves it all down. That's how we get water on one side versus the other. It doesn't happen very easily. Now, in the case of mitochondria, the way we get protons on one side versus the other is with the energy from foods. So if we're just looking at the mitochondria naturally, there was no energy, this would naturally equilibrate. So you have equal amount of protons or hydrogen ions on one side versus the other. Now, with our food, we get energy to push a bunch of protons on one side of the mitochondria versus the other, and that creates the gradient. So the reason we eat carbohydrates and fats is so that we can shove protons against the gradient up to where there's a lot of protons on one side and not another. And then it's the moving of the protons down the gradient back down that gives us this usable ATP. So here's our electron transport chain. So we have our ATP synthase. That's what shoves the ATP and phosphate back together. Now, also embedded within that inner wall of the mitochondria is what we call the electron transport chain. It'll have multiple complexes. We often just call them 1, 2, 3, 4. These are going to take in processed things from carbs, fats, and other things like proteins. They're taking the processed end products and they're going to come in and use some of the energy that's still left in those the processed parts of the carbs and fats. And that energy is going to power this electron transport chain machine. And it's going to give it enough energy to move the protons up here where there's already a lot of protons. And to do that, it uses the energy from food, it uses oxygen, and then it creates water. So the oxidative part of oxidative phosphorylation comes where, in order to shove the protons across the gradient up where there's already a lot of protons, you need the energy from the carbs and fats, and then you need the oxygen to facilitate that. If you don't have the oxygen, nothing happens. That's why if you suffocate, you die, because you cannot recycle. You can't get the protons up here, and then you can't get ATP to be recycled, and so you can't power your cells and you die. Now, this process is a little slow, lots and lots of steps, but it provides lots of ATP in a sustainable way, and it doesn't do it while polluting the muscle. It keeps the muscle relatively happy, where you don't have the acids and the phosphates all accumulating out by the actin and myosin, and so you can sustain this for a long period of time. You're going to take a lot of biochem classes and other things that focus on glycolysis and aerobic metabolism. For now, all that I care that you understand is that ATP resynthesis happens by having protons go down a gradient. And that movement of the protons down the gradient is what shoves the ATP back together. And then we consume carbs and fats and oxygen so that we can actually get the protons or the hydrogen ions up there in the first place. Okay. Now you'll take lots more classes, learn a lot more detail, but the consequences of this is that each of these bioenergetic systems have their strengths and weaknesses. The full breakdown of carbohydrates and fats, it's slow, but it gives you lots of ATP to power you for a long time. Partial breakdown of glycolysis is faster, but you only get part of the energy out of it, out of the glucose, and it pollutes the muscle, so it causes you to have fatigue. And then phosphogen system with phosphocreatine and adenylate kinase, these recycle ATP very quickly because they're just one step, but you only have a little bit of that stuff stored, so you can't do it for very long. That's all for today.